We're going to get the genetics vocab and the Punnett squares in this. So before we get going too far into this, I want you to make sure that you understand the concepts we're talking about. So previously we talked about DNA replication. We know that the DNA comes from the chromosome, and within the DNA is segments of genes. So within this segment here, if this is a DNA, it might code for a gene. And then we have alleles. Alleles are different versions of genes. So if these are a homologous pair of chromosomes, then in the same area on that chromosome, we might have coding for two different genes, which then passes on the traits. So the alleles are two different genes, and the combination of those two different versions impacts what our traits are. All right. So vocabulary. Allele is a form of a gene, and when we talk about alleles, we generally represent them with a capital or lowercase letter, like a big Y or little y. Dominant is a form of the gene, the allele, that masked others when carried by only one of the homologous chromosomes. So this is the one that's going to provide the trait. We use a capital letter for it, and it's the dominant trait is typically used when choosing a letter for a Punnett square. So if the color is yellow, we're going to use a capital Y. If the recessive color is green, it's going to be a lowercase y. So it's named based upon what the dominant trait is. Recessive, on the other hand, is the one that's essentially hidden. So it's there, but it's hidden if there's a dominant trait. The only way that the recessive trait is expressed is if there are two recessive alleles for the same characteristic. And that's with the exception of sex-linked traits. We'll get into that in a different lesson, so don't worry about that right now. So if green is the other color, we would use a lowercase y for that. Now here we've got a table that kind of shows some of the dominant genes versus some of the recessive genes. Clearly that got cut off, but you can kind of tell your differences from one to the next. All right, vocabulary continued. Genotype is the organism's genetic makeup. In other words, it's going to be the two alleles. So whatever the alleles are for the trait, that's what we're going to represent the genotype with. So big is going to be your dominant trait. Little is going to be your recessive, and then this is a mix of the two, which is still going to express the dominant trait, but it is a carrier for the recessive trait. So it could pass this on to the kids, to any offspring. Phenotype is the physical, notice that it's the physical appearance. Both of them start with PH, so hopefully that will help you remember what's what. And the phenotype does not always indicate the genotype properly. So we can have environmental factors that can affect a phenotype. Generally, genetics controls the majority of it, but the environment could also affect it. So here we have a genotype of big, we get a yellow color. Big, little, we get a yellow color because it's still got the dominant one, which is going to then hide the recessive, and then the recessive is the green. Now Mendel, when he did his crosses, he started out with purebred yellow and purebred green. So the purebred yellow could only pass on a capital, a big allele, and the purebred green could only pass on the recessive allele. So if you cross those in a Punnett square, which we'll talk about Punnett squares in a minute, you would only get a big and a little. So you'd get the big from the yellow one, you'd get the little from the green one, and you would only have offspring that would be heterozygous, which is big, little. We'll get to that term next. Okay, so and then we'll talk about what happens when you mix two of the heterozygous as well. Okay, so homozygous is going to be the same letters. It doesn't matter whether they're whether they are dominant or recessive, homozygous or purebred, just means it can only pass on that particular trait. So both alleles are the same. Remember, homo means same. Heterozygous is when the two alleles are different. So hetero means different, and you're always going to have a big and a little. Now we always write the big first. This one is written backwards just to show that the opposite parent could be little and big, vice versa, to show it on the chromosomes. But when we write them out, we always put our dominant letter first. A carrier is an individual that expresses the dominant trait but can pass on the recessive allele. So carrier and heterozygous are both terms that can be used to identify the big little. All right. Punnett squares are diagrams used to predict the probability of the offspring in a genetic cross. A monohybrid cross is a cross involving only one trait or characteristic. We're focusing on those to start. So this right here would be a monohybrid cross. We're only considering the trait that is represented by T, which is going to be a tall plant or a short plant. So when we do Punnett squares, you're going to have, we've got the parents are over here. So if one parent is big little, we're going to put a big here and a little down here. And if the other parent is little little, we're going to put little here and little here. And then all you're going to do is you're going to be putting the big T in each box going across. And then you're going to take this little t and put it in each box going across. And then you're going to take the ones from the top and take them down. And so we would end up having a big T from the left side here, a little t from the left side here, and then this little t is going to fill down. So you'll have little t and little t. So you'll end up with 50% that would be dominant because you got the big and big up here. And then these would both be recessive because all they have is the little little. Okay. And this gives us an example ratio. This does not go with the problem over here because it does not match it. If we were to come up with the genotypic ratio here, we would be saying we have big, little, and we have two of those, two of the little, little. Okay. And our phenotypic ratio for this particular problem would then be 2 tall, because 2 of them have the dominant, and 2 short, because 2 have the both little ones. Okay, using a Punnett square, determine the genotypic and phenotypic ratios of the offspring of a cross between 2 heterozygous tall pea plants. Now, if you remember back to Mendel started off with 2 purebreds, but when the 2 purebreds were put together, we ended up with 2 heterozygous pea plants. So we're going to have 2 heterozygous tall. So you'll have one parent that's going to be big little across the top and one parent that's going to be big little across the side. And then go ahead and cross these. So make sure you're taking your big one across the top and your little one across the bottom. And then this big one's going to go down and this little one's going to go down. And if you have any questions on what your results should be, you can always check in with me and I can verify whether you have it correct or not.